Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 41, where first I will discuss the monoterpenoids as component of floral scent volatiles, and then I will briefly discuss one metabolic engineering case studies where a gene of monoterpene synthase was expressed in a model plant with an aim to produce floral scent, and we will see what is going to happen upon expression of such gene. So, let us go to the concept covered. So, the concept covered is monoterpenoids dominate floral scent bouquet, that is our first part of this discussion, and then we will bring the issue of metabolic engineering. So, when we talk about the floral scent, although in this class I will only emphasize on the monoterpenoid component of floral scent, but to give the proper introduction about floral scent is this, that floral scent consists of multiple group of compounds. Okay. So, monoterpenes or monoterpenoids are one such group. Apart from monoterpenoids, the sesquiterpenoids are also present in the floral scent, and also benzenoids constitute a major portion of floral scent. In addition to that, aliphatic aldehydes, fatty acid esters, that also constitute the floral scent. So, floral scent basically a mixture of different components. So, that means that different metabolic pathways are operating in the flower in order to produce different scent components, and but the emphasis now I will only give on the monoterpene components, and when I will talk about benzenoids, phenylpropanoids, then there I will bring the issue of benzenoid scent components. So, just to fulfill your curiosity that how we study floral scent biology. As I said that floral scent, when we get the smell through our olfactory nervous system, that it is basically a mixture of multiple compounds, which could be as many as 50 or as low as 10 to 15. So, that means that when we it is important that we need to analyze the scent component. So, how to analyze that? So, GCMS is the major technique which is usually employed for analysis of scent volatile compounds. Okay. And next slide I will talk about how scent volatiles are trapped, but now. So, let us go to this concept. So, upon GCMS analysis we have we have identified there are multiple volatile compounds present, and based on that, for example, we get say B, we get C, we get D. So, these are the different volatile compounds. So, once we know this, once we know the chemistry of this, and we need to find out the logic or any relationship exists, if any, between these compounds. So, and based on our chemical knowledge, if we can theoretically predict that such relationship exists, for example, C can be formed from B simple by a hydroxylation, then there must be a hydroxylase enzyme which is carrying out the reaction. Okay. And then for example, in the next step, so that means C to D, D compound is basically a methylated compound. So, we can think of that there must be a methylation reaction or methyl transferase reaction is going on, which leads to methylation. So, methyl transferase reaction. So, like that way, and then once we know the structure of B, then based on our knowledge of the pathway, we can also make a prediction that B probably is formed from A, and A by another enzyme which perhaps chain shortening enzyme or chain cleavage enzyme is functional, and that leads to the formation of B. That means, by analyzing the compounds, it is possible to find out the relationship between these, and we can predict what sort of enzyme activities are there. So, once we know that, then what we need to do, you can get these compounds, synthetic compounds, and then you can carry out in vitro enzyme assay. So, in vitro enzyme assay, that means, you take a test tube, add the compound A, which is the substrate for the cleavage enzyme, and you make a protein preparation from the floral tissue, and then carry out the reaction in the test tube and see whether the crude enzyme, which consists of the chain cleavage enzyme as well, makes B from A. If it is successful, then it is fine. So, the next step will be, you add in the test tube B and carry out a similar reaction and see whether it makes C, and accordingly the reaction which constitute say a methyl transferase reaction. So, whether that exists or not. So, that way you have to add the C in as a substrate in the test tube. You may have to add a methyl donor SAM, and as a result of that, maybe the D will be, if D is produced, then you can be certain that such enzyme indeed exist. Once we know that, then it is possible to look for the genes. So, once we know the enzyme, so we can also already find out the what are the genes. So, the genes to be isolated. So, the earlier days, not earlier days, even decade back or less, people are using degenerate primer based gene isolation, because no sequence was available, and then you have to get the partial fragment, and then that to be cloned, and then full length fragment to be synthesized, and then subsequently it can be used for functional analysis, or those who are not interested in that, simply to study the expression, then from the core cDNA fragment primers can be designed and expression can be analyzed. But nowadays, with the advancement or with the cheap availability of the transcriptomics data, so the transcriptomics approaches are usually followed, and from the transcriptome data the genes are isolated and characterized. So, this is in general. So, that means, first compound, enzyme, gene. So, that is the classical concept, and the concept is still valid. Now, what I am showing here, this is basically called a floral head space. So, a live plant flower is inside. So, that is emitting volatiles, and this is a glass vessel. So, which is called a head space. So, and where scents are emitted by the flower, and this head space is filled up with that, and then it is connected to a vacuum through a vacuum pump, and in between one has to keep a filter what they have used. So, sometimes we call this as a column which will absorb the volatiles. So, if you attach it with a vacuum pump, and in between if you put this column, then what will happen? All the volatiles will be trapped in the column, which what is written filter here, and then later this can be detached, and the volatiles can be the mixture of volatiles can be collected in an organic solvent and later analyzed through GC or GCMS, and eventually you will get this sort of chromatogram, and with the use of library, NIST library, mostly the compounds can be identified, and confirmation can be done by running some standards. So, this is in general, and this is one real example in the basically in the laboratory. This is what is the head space, and this is a live flower inside, and here this is what I call the column, which consists of polymeric adsorbent, which will adsorb the volatile compounds, and then later it can be detached those compounds through organic solvent by passing through organic solvents, and then it can be concentrated and analyzed using GCMS to identify the compounds. So, as our point of discussion is on monoterpenes, monoterpenoids. So, I have mentioned in the previous class that linalool, a monoterpene alcohol. So, linalool constitute one of the major component of jasmine floral scent. So, this is basically a jasmine, this is jasmine flower, and the scientific name is Jasminum sambac. And so, this is the MAP pathway which is constituting the ultimately the monoterpene. So, the DXR, DXS are the two rate limiting enzymes, and here is the terpene synthase. So, terpene synthase is a big class of enzymes. So, where there are multiple terpene synthase enzymes exist, such as LIS stands for linalool synthase, BOS stands for osmium synthase. This is both are terpene synthase. So, beta osmium synthase BOS stands for. So, these are the major terpenoid monoterpenoid components of jasmine. So, that this was studied, and in order to know more about the biology of the scent emission, it is important to study the physiology of this. And just I would like to show you one time course analysis. So, this is basically the DXR which converts deoxydisallulose phosphate DXP into MEP. So, this is the enzyme, and the activity was measured throughout the floral lifespan, and you see the activity follows a typical rhythmic pattern. And similarly, different part of the floral tissue was also analyzed, and also the petal layers are also analyzed. Okay. And this is terpene synthase activity. Terpene synthase activity also follows a typical pattern. So, this is for the throughout the floral lifespan, and what has been found that petal tissue is actually is the place where maximum terpene synthase activity exists, and all layer contain terpene synthase activity, outer layer is little bit more, and this is basically a GCMS analysis where different components are detected. So, that means, when terpene synthase assay is done, so the GPP is to be taken as the substrate, and the flower petal protein extract was used as a source of enzyme, and this GPP, if it is able to make the products, for example, linalool and other components. So, that can be detected through GCMS analysis. Okay. Now, what I will show you a very interesting thing, as the flower remain alive for several days. So, what is shown here, that this portion is basically the night time, what is shown under the shade condition, and the portion unshaded are basically the daytime. What you see particularly for the linalool, that linalool maximum linalool concentration you find during the daytime, that means during the daytime compared to the night time, for example, the linalool. Whereas, in case of farnesene, although that is not our point of discussion, the sesquiterpene, it follows a different pattern. But if you again look into the geraniol linalool, so they are also maximum during the daytime. Similarly, another monoterpenoid which is nerolidol, that is also maximum accumulation was found in the daytime. Later, when we will study the benzenoids, there we will see that benzenoids accumulation actually reached its peak during the night time, because there is a relationship between the benzenoid emission and the pollinator visits, particularly the moth. So, but the terpenes, particularly monoterpenes, are emitted more in the daytime. So, such information is important, because if one want to study the gene expression or isolate the gene. So, it is important that which time point the petals to be used for such analysis. So, this is an overview, that is the subcellular storage and emission. So, this is the plastid where MAP pathway is operating, and of course, in the cytosol MVA pathway is operating. Okay. So, our point is only to see this now right now. So, it makes GPP, and then the GPP constitute the GPP basically serve as a substrate for monoterpene synthase. So, like terpene synthase, and it makes the monoterpenes including linalool, and these may directly emits, okay, or this can be stored in the vacuole. So, this can be stored in the vacuole either as free endogenous volatile, or we can call free internal pool, or this can be and this can undergo glycosylation, that is it linked with the glucose, and then it is safely stored there, and as and when required then there will be deglycosylation or glucosidase activity that leads to the release of the aglycone monoterpenes, and that eventually will be emitted. So, that means that there are three ways. One the monoterpenes produced it directly emits. Second is the monoterpenes can temporarily stored as a free monoterpenes in the vacuole, or for a long term storage they can link with the sugar and stored there. So, this is basically an overview of the different monoterpene components and how they are stored in a plant cell. And they are always low molecular weight compounds, lipophilic liquid, and the emission why it emits, because it is a high vapor pressure exists. Now, influence of temperature on biosynthesis and emission. So, one study was done with another Jasminium species, which is called Jasminium auriculatum, which is called juhi or juthi. So, these plants were grown in the plant growth chamber, which is basically the picture of growth chamber, where the 4 different temperatures were used. So, in one instance the plants were incubated at 20 degree, another instance 25 degree, another instance 30 degree, another instance 35 degree. Okay. And the plants were kept just at the time of bud formation. So, the buds developed and it bloomed, and then emission happened, and accordingly the volatiles were collected. This is basically the head space, and then it analyzed not only the emitted volatile, also the internal pool of volatiles what is present, that was also analyzed. So, what you see here that 30 degree is optimum for the maximum emission of linalool as compared to the other temperatures. So, even 30 degree temperature is slightly better than 25 for linalool accumulation inside the cell. So, from this knowledge, what is the practical application is this, that okay, growing the plant is growth chamber for commercial purpose is not feasible, but such study will throw you light how can you grow juhi or Jasminium auriculatum. So, you can use a shed net, you can use a plastic house, and you can maintain the temperature 30 degree by putting fan and cooling pad, and then you can grow there. And if you grow the plants outside where temperature variation is large, then you will be able to find out the difference in the concentration of the volatile components which accumulated when you grow at 30 degree and when you grow in the outside the field. So, it has a large impact because these flowers are used for different cosmetic application, not only that, even for the as a food ingredient, particularly it is mixed up with the black tea or green tea to make herbal, to make jasmine tea. So, it is important that the concentration of this compound should be at the higher side, and not only that, suppose if you want to make other products like attar and other, so from there may be different its uses in different cosmetic applications. So, there also, so it is always better to have a plant material with higher concentration of the desired metabolite. So, this is one way one can really make impact in the horticulture industry by enhancing the volatile terpene contents in the flower simply growing at a particular temperature, and which is possible. So, and so then there are some fundamental studies that as this terpene synthase. So, terpene synthase expression was studied at 4 different temperature. So, like 30 here particularly TPS there is not much difference. So, the gene expression level is more or less same, but what happens that beyond the expression level there is of course, the enzyme activity. So, that perhaps makes difference in the final production of the volatile components. This is what it is. So, it is not all that gene expression high gene expression makes the final product, it may not be. As you see that 20 degrees terpene synthesis is slightly less expression, but whereas 25, 30, 35 that is not much difference. Similarly, it is true for both the MVA pathway genes also. So, that means the post gene expression period is important or ultimately for which will determine the concentration of the volatile metabolites accumulation in the floral tissue. So, this is basically in brief about some studies done with jasmine with an aim to give some clue to the horticulture industry how can the cultivars or the farmers can grow jasmine in a more scientific way that can leads to more product accumulation and which will give a better profit to the farmers. So, that is an applied aspect. Now, we will go to more of the fundamental aspect, that is now we talk about the metabolic engineering application. So, the metabolic engineering of monoterpenoid pathway. I will try to explain in a very simplified way. First of all, what is the target? What scientists wanted to achieve? So, they wanted the first of all the source gene. Source gene is linalool synthase, LINA LOOL linalool synthase, LIS. Now, one information I need to tell you that linalool synthase is basically the gene was isolated from a plant called Clarkia brewery. It is a plant of native to South America, Onagraceae family. It produces big flower and the flower emit volatiles which is basically dominated by monoterpenes as well as by benzenoids. So, among monoterpenoids rather the linalool is one of the major dominating compound. So, actually that was the driving force behind isolating the genes of linalool synthase. So, before gene isolation they have also studied the pattern of linalool synthase enzyme expression throughout the floral lifespan, as I have shown in case of jasmine. So, here I would like to show you this in the form of a graph. So, like the level of activities, so level of linalool synthase activity in flower tissue throughout their lifespan. So, for example, Okay. This is -2, this is -1, okay, and this is 1, this is 2, and then this is 3, and so on. So, what is this basically? This is called time post anthesis. Post anthesis means that when the bud matures, it opens, and just it coincides with the bursting of the anthers. That means this is the anther. So, this is anther mature. So, when the anther will burst, that is called dehiscence of the anther. So, that leads to and where you will see that pollens are released. These are nothing but the so this happens the phenomenon called anthesis. So, time post anthesis means that at 1 it is the time when the anther bursts and it releases the pollen, and that is the time also the petal opens. So, that means the bud matures and it fully blooms, and at these are the points where the linearol synthase activity was measured. So, linearol synthase activity was measured. So, like I will put the scale y axis is for the activity. So, the LIS activity. So, and that may be a unit called picocat or nanocat flower tissue. So, that anyway I am not talking in that details. So, what I am going to show here is this, that how the linearol synthase activity behaves. So, this is 2 and then this is 1 and then 1 it goes slightly down and it goes further down 2 and then now if you join this point, then you will find that so that means the maximum linearol synthase activity we will find at -1. That means just the day before the opening of the petal. Okay. So, that means the petals are more or less matured, but the bud is still closed, and that is the point maximum linearol synthase activity was detected, and 1 is basically when the 1 day 0 means at the time just opening, 1 is the day just the anthesis after the anthesis. So, it is basically getting down. So, this is typically the pattern of linearol synthase activity. So, this is very important. So, to know that how this terpene regulation happens, because it will be also interesting to see that how this differs in the different plants, how this differs in the different plants. Okay, I just made a small leave it like that. Okay. Now, this is what it is, and next point is this, that this source, what is the source of this is linearol synthase from the Clarkia. So, if you go to the Next slide. So, the source LIS from Clarkia brewery, and now the target plant they have chosen Arabidopsis thaliana. So, Arabidopsis is basically a model plant used in plant biology, okay. And so they transfer the linalool synthase gene from Clarkia to Arabidopsis. So, the method what was used is simply agrobacterium mediated transformation. So, that means the linalool synthase gene is here. So, it should be under the control of a promoter and a terminator. So, terminator is added here and promoter is here, but in between the promoter and terminator what needs to be added is basically a transit peptide sequence. And why? Because this is the sorry, this is the promoter, right? Okay, I will. So, why transit peptide? Because what I have said in one of the previous classes, that the monoterpene pathway is operating inside the plastid, and this is basically we are transforming the gene into the nucleus. So, the through agrobacterium mediated transformation. So, that means using A tumefaciens the gene was transformed in Arabidopsis, and it is integrated into the nuclear genome. From there transcription will happen and it makes the linalool synthase messenger RNA, and then finally linalool synthase protein. But if you do not put the transit peptide, then that protein will be there in the cytosol, and if it is there in the cytosol, it will not get the substrate, because remember the GPP is the substrate and GPP is basically localized in the plastid. So, therefore, the transit peptide sequence was added, because that once the protein is produced after translation and it should contain the transit peptide at the N-terminal region. So, it will drive that protein inside the chloroplast, and then it will be able to function. So, transit peptide adding basically driving the protein into the chloroplast, what is the site for the enzyme activity. Okay. So, that is what has been done. Now, question is that what is the result? Result is this, that they have successfully produced transgenic plant, Arabidopsis plant expressing LIS gene. Okay, gene expression was confirmed, everything. But what happened, that when they try to measure the linalool from the floral tissue, unfortunately no linalool was detected in transgenic Arabidopsis flower, where LIS was expressed. So, in fact, LIS was expressed constitutively. So, the constitutive promoter was used, that means throughout the plant linalool synthase was expressed. But anyway, it is expected that linalool is mostly is a monoterpene volatile, as the plastid is the component. So, they check first from the flower, or later also they check from the different parts, but no linalool was detected. So, then why no linalool is detected? Although the gene expression worked very well, so that means the gene was functional and the substrate GPPs should be plenty available. So, why it is not? That is what is the question is. So, then they try to find it out what happened. So, then they did a very careful analysis. They have made extract from the floral tissue as well as the vegetative tissue, and what they found that it is not linalool, but basically they have this time they detected detection of S linalyl beta D glucopyranoside. So, this is what they have found in the tissue. So, as the name indicates, it has some relationship with the linalool. So, what sort of relationship? It appears that linalool perhaps produced and then it undergoes glucosylation, leading to formation of this compound, which is non-volatile and which is stored in the vacuole. Okay. Then what they did basically, they try to get the linalool out. Simply they treated this with beta glucosidase. So, beta glucosidase what it will do? It will cleave the glucose out. So, now what you will see that linalool plus beta D glucose. Okay, I put beta D glucose for better writing. Linalool plus so glucosidase treatment in the tissue, now leading to detection of linalool plus beta D glucose. Okay. So, that means it is clear that LIS, that means linalool synthase gene was successfully expressed, the gene was function, it took all the GPP available in the tissue and converted into linalool, but the plant found linalool to be a strange molecule because it is not meant to produce linalool. So, immediately what happens, that glucosyl transferase is induced, and as a result of that linalool is stored as linalool glucopyranoside. Okay. So, what happens, that this is because of this induced glucosyl transferase endogenous. And this is the transgenic LIS or LIS gene, we can say. Transgenic LIS gene means that the gene was basically transferred from a different plant. So, okay, better we use the term LIS gene from this what is the Clarkia species LIS gene. So, this is what is the outcome. So, that means plants were successfully producing linalool, but converted into linalool glucoside, and that is maybe a sort of detoxification mechanism or some sort of adaptation. So, that means the message is that whatever you want to do with the plant may not do, they are also equally clever. They will do whatever they feel that which is necessary for their proper survival. So, this is a nice work, and this paper basically published in a famous journal which is Plant Journal or The Plant Journal. Full name is The Plant Journal, in the year pretty old 2001, volume 27, page number 315 to 324. So, what authors have written, that linalool produced by the plant in various tissue is not present as free linalool, but efficiently converted to non-volatile linyl glucopyronoside by the action of endogenous glucosyl transferase. So, what I have said that this is induced. So, the result present demonstrated that yes, monoterpene production can be altered by genetic modification, and that the compound produced can be converted by endogenous enzyme activity. So, there is the challenge. How can you really make the plants to produce free linalool? It should not get converted into a glucoside, that is the challenge, and that is the difficulty with the plant system. So, with this I end this class. Thank you.